You're welcome, sir.
We have been in all our trainings. We always try to keep to our time, but today was inevitable. Well, so I'll quickly share my screen so that, yes. Once again, I want to say thank you for being around, and my phone even went off at the same time while I was trying to hurry up. So for those who might have tried our line and it was off, we are really sorry for that. So I want to share my screen quickly. If you can see my screen, just let me know. Can you see my screen?
Yes, sir. We can see it, sir.
All right.
You can see your
So
screen.
we thank God for today. Okay, a quick one. This was where we stopped. We able to convert our natural products into their SMILE code using Open Babel, and from there we took the SMILE codes to Admet Lab, which we said we are going to do that in batch by batch, due to the traffic in the site. Because if you upload up to 1,000, because the site can hold up to 1,000, but if you upload so large, it will give you an error message. So it's better we do that gradually. Then later you now compile all the results together. So I believe that within the weekend we must have tried something with our own data, with your own data. Because I made it clear that try using your own data to do your. Okay, so we wanted to now screen those compounds based on their toxicities. There were a lot of toxic profiles we got. We got a lot of toxic profiles. Let me, if I can still have that data. Let me open up that data so that we're going to use it right away. So we had a lot of toxic profiles, and where we stopped was we wanted to now look at the implications of those toxicities, those toxic profiles, those toxic parameters. We wanted to look at their implications. We hope you can see my screen, right?
Yes, sir.
All right, so we started from here. So from BCF, this is where toxicity starts. From BCF down, as we are going, all these ones are toxic profiles from this BCF. So starting from this BCF, all these are toxic profiles. So, and we said that Admet Lab gives toxic profiles a value of probability from 0 to 1, and that's where we stopped. We wanted to know the implications of those values, those toxic parameters, their role. Because if we say let us do them in the lab, we're going to spend much money to do each and every one of them in the lab. So, but this has been modeled so we can just assess them. So we have done this part where we said that a toxic value, a value between 0 to 0.3 it implies low risk and safe. Then toxic value between 0.1 to 0.31 to 0.5, we said it is moderate risk, which calls for caution. Okay, so you just be careful while you are working on with those values. And then a result of 0.51 to 0.7 is high concern. So there's a need to modify the structure of those compounds if we are really interested in those compounds, and unfortunately they happen to give a high risk value. So it calls for a structural concern. That means we should work on the structure so we can optimize the structures of those compounds. Maybe your interest is not to formulate a herbal product from the plant material. Maybe your interest is to identify a lead that you can synthesize and continue your in vivo work. So if that is your aim of getting a lead from this natural product, it means that if that your lead happens to have high risk of toxicity between this 0.51 to 0.71 in any of the parameters, it calls for structural concern. So you have to modify the structure, and you can use the little idea we shared on Pharmacology. You can use that idea to do that. You can use that idea to modify it. Then when you have a toxic value between 0.71 to 1.0, very high risk. So that's the likelihood for toxicity. So we are saying that such range of value, there's a likelihood for toxicity. You know, this is probability. So there's a high probability for toxicity between 0.71 to 1.0. So we say that these ranges guide us if we are to go or not to go. So it will guide your go or no go decisions in drug development. If we are to continue or not, which we concluded last week that even though we're having red flags in these compounds, that it now depends on the synergy, the other plants we are using in combination, because you don't formulate a herbal product with one plant, so that the effect of the other plant you are using might, that is probability uncertain, that might help inhibit this toxicity, or even might skyrocket, might increase the toxicity. So it's a probability aspect. But once your results of your natural products is giving you toxic, a good percentage is toxic, just be cautious. Just have that consciousness that you have a lot to do. So we started by saying that we have about 24 toxic profiles we want to release to you, and we don't just want to screen them without you knowing these toxic profiles and their implications. You know their implications, what they imply, what they do. So number one is HERG. So we shall look at the meaning of HERG. So HERG, the meaning of that HERG means human eta-agogo related gene. Human eta-agogo related gene. Now, what that means is that this gene is usually found around your kidney. It controls homeostasis, is in charge for potassium channel and balance. And, this potassium channel and balance plays major role in cardiac, in cardiology. It helps to determine if you have this tendency of having a cardiac arrest or not. So it controls cardiac repolarization. Now, what does the result tell us? The implication of this H inhibition. So if your compound was able to inhibit this H, that means it will cause prolongation and fatal arrhythmias. Now, compounds with high range of high H risk often fail clinical trials. So your compound should not inhibit this gene, because this gene in some way helps in potassium channel and control potassium balance. So if your bioactive compounds happens to inhibit this gene, that means that compound is toxic. So the range interpretation for this H 0 to 0.3 implies low cardiac risk, acceptable. So when you have a bioactive compounds that the value for this H is between 0 to 0.3, that means it means that compound can be accepted for H, so there is low cardiac risk. But if it's between 0.31 to 0.5, there is moderate risk. So we need to monitor closely. So we just give you 3 range. We are giving 3 range. I have to summarize them to 3 range from 0 to 0.3, then average of 0.31 to 0.5, and from 0.51 to 1. So now this is to tell you that if you are establishing your benchmark, I think that the best benchmark should be no more than 0.3. So if we can achieve a good number of compounds that we succeed on this range, is a good thing, is a good result. But I doubt, I'm afraid, if we could, but it all depends on your data. So we shall do that. Now another profile that we shall look at is the H, but this H is assessed in a concentration of 10 micromolar. So here we are considering the inhibition at a concentration of 10 micromolar. So could this compound inhibit this gene at a very low concentration of 10 micromolar? So the meaning is that there's a likelihood of H inhibition at a screening concentration of 10 micromolar. So we are looking at the likelihood of inhibiting this gene at a concentration of that compound, if that compound should be induced at a concentration of 10 micromolar. Let's see the ability to also inhibit this gene. That is the second properties that we shall look at. And the implication indicates early cardiotoxicity during dose escalation. So it will help us to see that at low dose, even at a low dose of 10 micromolar, there is toxicity, there is cardiotoxicity, because we always claim that at lower dosage, there's always low risk. There is a low risk at low dosage. But at a higher dosage, there's a probability of encountering high risk. So that is why this parameter is not assessing the cardio arrest, the cardiotoxicity at a very low concentration of 10 micromolar. So if it happens that the value of that bioactive compounds for this parameter falls within 0.3 to within 0 to 0.3, it is safe at screening dose. Then 0.1 to 0.5 implies potential dose dependent risk, because this parameter is based on dose. So at 0.2 to 0.5 implies potential dose dependent risk. At that point, you have to look at the dosage. Of course, anything above 0.5 indicates strong cardiotoxic signal. Strong cardiotoxic signal. So we should always avoid toxic values between 0.51 to any toxic value above 0.5. We can equally manage below 0.5, 5 and below. But the best, if you have to set your benchmark, put it within highest, not more than 0.3. If you can achieve compounds within that range, very good for you. Number 3 is DILI. What is DILI all about? It means blood-induced liver injury. That is DILI. Is among the parameters you see in ADMET lab, which I believe that most of us have even seen it, we have done it. We have seen those toxic parameters. So it's just for us to know what those values mean, what those parameters mean, and the implications. So DIL is a probability of drug-induced liver injury. So it tries to check if liver toxicity is a major cause of a drug withdrawal. So here I want to see if this drug, why induced, can cause liver injury. That is what this parameter is looking at. Is there any probability that this compound can cause a liver injury? If I induce this compound, if I take in this compound as a herbal medication or as a pure compound, the presence of that compound in this herb, in my formulation, will it cause liver toxicity? So, and it is a major cause of drug withdrawal because liver, kidneys, they are vital organs. They are vital organs. So your drug must not temper these organs. So if your value is below 0.3, that means you are having low hepatotoxicity. So you're having low liver toxicity based on the parameter of DILI. If your value is from 0.31 to 0.5, that means moderate liver risk. So there's liver risk, but it is moderate. But when your value is always below 0.3, below 0.3, that means you are totally safe. So if you are lucky that your extract of the formulation, a good number of the bioactive compounds are within that range of 0.3 and less, that means you have skipped that major aspect of drug withdrawal, which is the aspect of the safety. So we already know from 0.51 to 1, high liver injury risk. So that means that such a compound will damage your liver. So these results, these are computer-generated results, but they were generated not just by computer, but they have been generated based on experimental data, which was used to model the algorithm that generated these figures. So these figures, they are approximately true values, up to 90% that you can trust them and rely on them. Number 4 is the AMES. The AMES means the role is for mutagenicity potential. So it's based on mutagenicity. So what the result implies is that AMES positive compounds may cause genetic mutations and cancer. So when you have a compound that is highly toxic for AMES, what that means is that compound with a value between 0.5 to 0.1 from the AMES property, that compound has the potential of causing a genetic mutation and cancer. So that compound is carcinogenic. So you must be aware of this. So it is a careful screening. It's a very, very careful screening, thorough screening to screen all of the bioactive compounds for all the parameters. So we screen the bioactive compounds for all the parameters at the same time. So we can get good compound. It can even change your objective from the formulation to getting a lead. If you have seen that, out of 500 compounds, 450, out of 500, 450 are toxic, then that 50 non-toxic is giving you a good result that you can move with this 50 and discover a lead from there, and that will be a good innovation. So 0.0 to 0.3 are non-mutagenic, so it does not cause any genetic mutation. 0.31 to 0.5 weak mutagenicity, like moderate. 0.51 to 1 strong mutagenic risk. So when you see these values in those results, you now know what they imply. We have ROA, which also implies the route of administration toxicity. The route of administration toxicity. That is what the ROA talks about. Now the meaning is that the meaning here is toxicity associated with the intended administration route. So want to see, could there be any toxicity based on the pathway of administration? the route of administration based on the way that compound will be administered to the body. Could there be any toxicity? So the implication here is that ROA helps to determine formulation safety and patient exposure risk. So here it helps to tell you that your formulation, the way it will be absorbed, if there is a possibility of affecting the body, causing adverse effects based on the mode of absorption. So when your value ranges from 0.1 to 0.0 to 0.3, that means that the route of absorption of that compound is safe. So if the essence is once you see that this route of absorption is toxic, then you have to now start looking for a means that compound can be administered to the body without being toxic to the body. So you start looking for alternative way. But once your ROA gives you safe, it means irrespective of the route of absorption, irrespective of the medium you want to take the drug to the body, that compound is safe. That compound is safe, irrespective of the method of administration. That is what that values of 0.0 to 0.3 all implies. We already know that 0.5 to 1 point means unsuitable for intended route. So you have to look for a route that will not be toxic. That is a stressful word, though. Number 6 is FDMDD. Here is based on the FDA approval. So this is the likelihood that safe exposure exceeds FDA maximum daily dose limit. So here it gives you a value if the compound can easily be approved by FDA. The impression is that it affects dosing strategy and therapeutic window. So if your value is if your FDMDD is between 0.0 to 0.3, that means safe daily dose. So what that implies is safe daily dose. Please, if the mic is on, just help me to mute the mic so I can hear myself. 0.31 to 0.5 means narrow dose margin. I'm getting echo. The source. Hello. Okay, thank you. So I'm taking to a journey now that you just have to be careful and you just have to listen because they are much, but we must take you through them so you know their implications. You just have to know the implications, and it really help you in designing your benchmark. So 0.5 means dose limiting toxicity. Then we have the screen sen, which is the screen sensitization. This is another toxic profile from ADMET. The screen sensitization is the probability of inducing allergic skin reactions. So with this compound actually induce skin reactions. If I take in this compound, will I have skin reactions? Just like what we know about chloroquine, that some persons are allergic to it. If you take it, it begins to scratch your body. So would there be any skin allergy from that bioactive compound? So these parameters will help you to explain that. The implication is that import is very important for topical and transdermal drugs. So mostly drugs you want to use on the screen. Drugs you want to use on the screen. So you look at these parameters for those kind of drugs, so that you will not take an ointment, a cream, or tube that will then bring to cause allergy to the skin. And mostly some of us who are interested in antifungal drugs, you know that most of the common antifungal therapies are always applied at the surface of the body as cream, as ointment, as tube. So if you have that interest in doing a work on antifungal, that means you must take note of this skin sensitization. So the value between 0.0 to 0.3 means sensitizing, that means it's okay. 0.31 to 0.5 mild sensitization. Then 0.5 above, strong allergic risk. So always on away from 0.5 and above. Always on away from 0.5 and above. We can equally manage 0.5 below, best 0.3 and 0.3 below. But anything 0.5 above, you must take precautions about that. Now, this is carcinogenicity, cancer causing potential. Does that compound have a cancer causing potential? Can it cause cancer? It's just similar to what we have looked at before. So does it have that potential of causing cancer? That is the carcinogenicity. And the implication is that carcinogenic compounds face regulatory rejection. Once your compound possesses this carcinogenic potential, it will automatically be rejected. It will be rejected immediately because you are giving us a drug that will cause cancer tomorrow. So we just reject it. So the same thing, 0.0 to 0.3 means non-carcinogenic. 0.31 to 0.5 possible risk. So to me, I prefer working with 0.3 below, where there is low risk, very safe. And even if you go to the wet lab to verify, once your in silico is telling you that this compound does not have that risk of toxicity, even if you go to wet lab, you still have that same kind of result. Yes, that same kind of result, that compound is safe. Then you know that 0.51 above means high cancer risk. So you don't even go there. Another one is the EC, which is the eye corrosion. The eye corrosion. Its ability to cause irreversible eye damage. So there are some drugs you take in, you absorb, those drugs will have a tendency of interfering with your eye glands, and it will cause eye damage. So we can also look at that toxic profile. Now, the impression is that this parameter is very much critical for aerosols and ophthalmic drugs, so drugs that are used for the treatment of eye. So there's a work I did at Futo for a conference at Futo. That work was on glaucoma. And last year I did a work on cataract. So if those work happens to maybe at the end of those work I happen to get a good compound from those results, it means that compound I must check if it's possible to cause eye damage. So the implication, the range, still what we know: 0.0, 0.3 non-corrosive, 0.3.1 to 5 moderate damage risk, above 0.51 severe eye damage. So always on away from any toxic values between 0.51 and above. Always run away from them. More than half, anything more than average is a failure. Above average is not accepted. So if a failure above average is not accepted, then so it should be aware of that. EI. So if you see EI means eye irritation. That is EI. EI means eye irritation. Yes. This might look like I'm just talking, but I have to do that. Just have to do that to help us and guide us. So EI is eye irritation. It is a potential. It means that compound has the potential to cause irreversible eye irritation. So eye problem. Don't produce a compound, don't produce a drug, a herbal product that will cause eye problem tomorrow. There is this ideology we have in herbal product. They said it will cure today and cause something else tomorrow. Is because we don't screen them properly based on their toxicity. That is where the problem always arises. The representation is that if your compound has EI, high value of EI, it will influence labeling and formulation. So it has to influence your labeling and mostly your formulation, because such a formula will definitely lead to eye irritations. You keep scratching your eye. Of course, we know the range 0.0, 0.3, non-irritant. So that means non-irritant is okay. 0.31 to 0.5, mild irritation. So always run away from 0.51, severe irritation. Even most times, I even run away from 0.5, 0.4. Most times I run away from them. The next one is respiratory toxicity, number 11. Respiratory toxicity. So we are looking at for the it is the potential of that compound to damage lungs or airways. Does it have effect on my lungs or my airways? Does it have effect on my breathing pathway? So you shall look at that toxic potential. You see that if you want to do this in a lab, respiratory toxicity, whoever that will design that work must be an expert, because you're going to wonder how I don't achieve it. But with the help of in silico, it is a good one. So if your value happens to have high value, high respiratory toxicity, it limits inhalation and aerosol use. So most importantly, it will limit your breathing, it will limit inhalation, and you know what that means. Of course, the range 0.0 to 0.3 is respiratory safe. So anything 0.3 below is okay. 0.3 to 0.5 moderate is moderate. 0.5 implies pulmonary toxicity. So don't near there for this compound, for this profile. The next one is HHT, which is human hepatotoxicity. So it's similar to that of liver, but this want to see if the compound has direct impact on liver cells. So here we are looking at liver cells. Previously, DILI was liver injury, but here it does it have effect on liver cells. So if yes, that means there will be a confirmation. It's already a confirmation on a reward liver safety risk. So there is high liver safety risk if your compound happens to have a 0.5 and above for HHT or HHT. So below 0.3 is safe, below 0.5 to 0.31 mild toxicity, 0.5 to 1 severe hepatotoxicity. So be careful of that. Number 13 here is neurotoxicity. Here we are looking at the potential for nervous system damage. So this compound has ability of damaging my nervous system. This is mostly when the compound was able to permeate the brain barrier. So the BBB, the blood-brain barrier. So it has the ability to penetrate this blood-brain barriers. That means it has effect on your nervous system. So you be careful of that. It means that if your value has high probability score for neurotoxicity, it means undesirable or useless CNS targeting is intended. So your CNS will be at risk. And we know that if the CNS is at risk, then the human body will not function, because it will operate by impulse and signal. So that would be a challenge. The range, we already know the best is 0.3, which is neural safe. 0.3, 0.1 is moderate CNS risk. 0.5 above, neurotoxic. So that means it can cause neurotoxicity. Number 14 is autotoxicity. Yes, number 14 here implies autotoxicity. So what we mean by auto, it has to do with your hearing ability. So risk of hearing or balance impairment. That is what that aspect implies. This autotoxicity, the auto system, which is ability of hearing and balancing what you hear. So if your compound has high value for autotoxicity, that simply indicates serious adverse effects, especially for antibiotics. So antibiotics have this trace of causing a autotoxicity effect. So you must take note of that value. The range is already clear with us. Another one is hepatotoxicity. Does it have the potential of causing blood toxicity? So can it affect my blood cells? Can it affect my white blood cells? Can it affect the red blood cells? So these are what these parameters cause for. That is what the hepatotoxicity is all about. So you try to know if the value is high or low. Of course, if the value is high, it means it can cause anemia or immune suppression. Of course, if that compound gives a high value of hepatotoxicity, that implies that it would possibly reduce the level of your white blood cells and your red blood cell, platelet, and other blood cells. And when these blood cells are altered, then you are giving room for immune suppression. So we should always take notes of that toxic red flag called hepatotoxicity. Below 0.3 is blood is safe. Below 0.5, mild toxicity. For 1 below, severe hematological risk. Run away from this range. The next one is nephrotoxicity. So I'm just compiling all the toxic profiles, and that is really what we need to know. Nephrotoxicity, that is for kidney. Does it have potential of causing kidney damage? Kidney damage. So instead of you to be doing your wet lab, and for those in biochemistry, you do effect of a liver extract on kidney function parameters. Some work on liver function parameters where you do your AST, LLT, ALP, and all those parameters. So persons willing to do parameters like creatinine level, urea level. So these parameters, potassium and sodium level. So these are parameters that we don't need again for you to do them in the lab. You can generate your result based on the prediction. So if your compound happens to have a high result for high probability value for nephrotoxicity, that implies that there's a limit in chronic or high dose use. So you should avoid using a high dose so that it will not destroy our kidney for us. You already know the range. No need to talk about the range. The kidney safe, moderate renal risk, and nephrotoxic, that is 5.1 to 1.0. Don't worry, we're almost coming towards the end of this aspect, so we can do some hands-on. We have genotoxicity. So you see, where science has taken us to very beautiful, very, very beautiful. We have genotoxicity. That is for DNA damage potential. Does it have impact on my DNA? If the value is high, that means it implies that compound is strongly linked to cancer development. So it can easily cause cancer also. So you should take note of genotoxicity. We already know that below 0.3 safe, moderate, and high genetic risk. 0.51 to 1.0 is showing us high genetic risk. We have the LPMI, A22C. Now, all these ones are cell-based. So I've mentioned these 17 are very important. The 17 I just mentioned are the most 17 that you should not joke with. Even if you don't want to select all the parameters, but you must not joke with this 17. The 18 down, they are cell toxicity as a result of interaction with some cells. So this one looks out for cytotoxicity against cancer cells. So this one is usually for cancer cells. So when the value is high, it means high values it indicates anti-cancer potential. So this one now is showing that this compound can inhibit your cancer cells because they are cancer cell lines. They are cancer cell lines. So you can help to know if there is a risk if it can inhibit a cancer cell or not. All right. So of course, below 3.0 is meaning that that compound does not have the tendency of attacking the cancer as an anti-cancer potential. But if the value is from 0.5 to 1, that means that compound will possess strong anti-cancer activity. Another one is the A549, still for cancer, lung cancer cells, still for cancer cell line. So the meaning is it measures lung cancer toxicity, and the result implies that high value of your A549.
Hello, sir. Hello, sir. Good day, sir.
Yeah.
Please, I want to know, is it anti-cancer activity the high value or carcinogenic? Is it carcinogenic or anti-cancer? Because when it is high, it means...
Yes, this was now anti-cancer.
Anti-cancer, not...
Yes, against cancer cell lines, yes.
When it
only
is
from
high.
here, from this 18, from LPM 822C and A549. So these ones are not really considered as toxicity. We bring them just to see if it has effect on cancer cell lines. So that's why I said that you concentrate on the first 17. From number 1 to 17, the list are the ones you pay attention to. This ones is just to see if these compounds has impact on cancer cell lines. Of course,
I know I'm trying to, I'm looking at the value here. When the value is high, it means that it has a lower ability to cause cancer. Is that what it means? When the value is high.
Yes.
When the value is high.
The reverse now, good. When the value is high, it means ability.
It has a lower ability to cause cancer.
Lower value means it can cause cancer or does not have impact on cancer cells. But high value means strong anti-cancer activity.
It's okay. It's okay, sir.
Fight against cancer cells, right?
Yes. So even HEC 2932 is a cancer cells, but related to normal kidney cells. So all these ones, we don't regard them as toxicity, but they are included from the ADMET results. So I just have to bring them in. But our decision will be based on the first 17. Based on the first 17, because those ones are safety parameters.
Okay, because I was looking at from 18. Because I thought if they are included, they are not toxic per se. They are not cytotoxic per se.
Yes. So we are going to look at even this 21, this BCF. These BCF are for environmental concern. So from 21 to for maybe you want to produce a pesticide, insecticide, something you're applying in the environment. That is where we look at this BCF is for biocontrol factor, concentration factor. So we may not look at those aspects. Maybe where you may just look at BCF is where they are disposing this drug as waste, because drug disposal management is a factor you consider while you're making your drug. If we dispose this drug, this my formulation, if you happen to dispose it, does it have effect on the environment? If it discharged on the aquatic system, does it have effect? So that is where the BCF comes in. So you can still include it, but most importantly, you build your model using the first 17 that we explained. Here is a probability that a compound may accumulate in living organisms relative to its concentration. So the bioaccumulation potential of that compound, and that compound might be accumulated in living organisms. A high bioaccumulation increases long-term ecological toxicity and regulatory concern, especially for chronic use drugs. So that, but this bioaccumulation is not really for humans, but it's mostly for the aquatic organisms, and not really assessing how it's bioaccumulates in humans for a long time. If you want to look at how it bioaccumulates in human, then there is this excretion potential called half-life, T0.5. So that is what you look at. So probably I shall explain the half-life of a drug when we get to pharmacokinetics, because it's a parameter under excretion. T0.5 is a parameter under excretion. So the implication point below 0.3, that means low bioaccumulation, which implies that it's safe environmentally. 0.3 to 0.5, moderate accumulation. 0.5 to 1, high bioaccumulation, which implies environmental risk. We have another environmental factor, which is IGC50, inhibitory growth concentration at 50%. This concentration is required to inhibit 50% growth of aquatic organism. So we are looking at the concentration that can inhibit the growth of aquatic microorganisms at 50%. It reflects environmental toxicity that may disrupt aquatic ecosystem. So does it have the ability to inhibit the growth of these aquatic microorganisms? If the value happens to be low from 0.3, it means low environmental toxicity. That means it doesn't have impact on these aquatic microorganisms. But if the value is high, it means that there is high ecological risk. So it has impact at 50%. It has impact on the aquatic microorganism, which is not supposed to be so. We have the LC50DM, which is the latter concentration at 50% for Diphaenia marginal. So all these ones are not necessary for us, but I just want to show you, because it's part of the toxic profiles in ADMET. So this looks at for the probability that the compound causes 50% mortality in Diphaenia marginal. The import is very important for environmental regulations. All this is so I will do more on this area next month, February, when we are dealing on green chemistry. That is when I will really emphasize more on the implication of this environmental toxic red flags. So I will spend my time on these aspects by next month training on February, which is on green chemistry. We shall look at those aspects. So on that note now, all these ones are not necessary for us. I just have to bring it in because of they are found in ADMET. So in conclusion, natural product toxicity screening is indispensable, is an indispensable component in modern pharmacological and biochemical research. Like I said before, don't do a work without screening your natural product for toxicity. Don't try it. Don't do a work without screening your compound, whether you use natural product or whatever. Don't do a work without screening them for toxicity. Now, with the integration of in silico toxicological prediction, researchers can now systematically identify safe and effective bioactive compounds for medicinal plants, which is what we are entering right now. Identify those compounds that are safe and effective bioactive compounds. So it's two things here. Once you are safe, then there's a tendency that it could be effective as a bioactive compound. Yeah, but we will still do a screening on how to screen a bioactive compound, identify the ones that we actively bind before doing my docking. There is a technique which you can identify the ones that could actively bind from your natural product, and that is why we brought that module for natural product mining. That one is actually lead mining for natural products. So without me going for docking, if I have 100 bioactive compounds, I can easily know the ones that have the tendency of giving me effective binding that would give an effective result. So that's what we shall screen also. And after that, the remaining part is so easy for us to walk through. So as herbal medicine usage continues, which is our goal, as herbal medicine usage continues to rise globally, rigorous toxicity screening ensures that natural products transition from traditional remedies to scientifically validated therapeutic agents with acceptable safety profiles. So as herbal medicine usage continues to rise globally, we are ensuring that rigorous toxicity screening. So if we can screen those herbal medicine rigorously, then we are ensuring that our natural product transition from traditional medicine to scientifically validated potentials will be well acceptable with good and acceptable safety profiles. So it's systematically, because you do that consciously. So at this point, we have taken all the toxic profiles. So let's go for the screening.
Before we go for the screening, can we ask questions?
Let's go back to what I said. But then,
yes. Can we ask questions? Yeah. I have three quick questions. One, among these parameters on the ADMET lab, which one can be used for reproductive toxicity? Yeah.
Reproductive toxicity. Okay. Yes. Okay. Go ahead.
That's one. The second question is that, as we can see, there are some features whose scoring does not follow the 0-1 system. By the time we are interested in those features, are we going to be screening them individually, unlike those that have 0-1, where we can group together? For example, I'm interested in one of these parameters that does not follow that metrics system, 0-1.
Like which one is that?
Well, let's say from your, what's it called now? There are some of them that don't have, maybe on this window, we don't have something like that. But there are some of them that don't have a 0-1 metric system. I can't.
Yeah, but not for toxic, not for toxic parameters.
Not for toxic parameters. There are other parameters that don't have 0-1 system. If you are interested in those parameters, are we not going to do them singly, because we may not be able to combine them together as we are combining the toxic parameters? That's the second question. The third question is this: even within those that have the 0-1 matrix, there are some of them that their directionality are opposing each other. Let's take, for instance, I'm interested in toxicity where 0 signifies good, 1 signifies bad. If I want to combine that with another parameter like permeation of the blood-brain barrier, the metric there is opposite. Drugs that permeate the blood-brain barrier very well, we have a score of 1. Those that poorly permeate, we have a score of 0. By the time I want to compare, I want to combine, let's say, blood-brain barrier permeability and toxicity, then it's difficult for me to do that screening together. So what do we do in that case? Do we also do them separately or what?
Okay, where do I start from? Let me start from, let me go, let me use our pathway, retrosynthesis. So I will go from behind. Now, in this case, you wish to see if you can combine two parameters that are not on the same category, like the BBB and the toxic parameters. So once you have seen that they are opposing each other, so you don't go, you can't combine them, because you can only combine them if their criteria are the same. So what you will do on that note is screen them separately. So you can screen them separately. Probably you first screen on toxicity, and the ones that passed, you now screen for the BBB. Yes, you screen for BBB. I would recommend, why I will not go much. I will not go much is because of the scope of the training. Yeah, the scope of the training. But then we just released the training today. We launched an advert today, this evening, on our free training coming forth by first week of February. That is Bioinformatics. Now, although we did Bioinformatics in this training, but we just did that in line with macrognosy. We did not broaden the scope of the Bioinformatics, because only Bioinformatics is another course on its own. But we just took the part that concerns us in this training. So, but in that 2 days training, that workshop, they're going to expose you to up to 10 database that can even help you in doing this training. So we will not tell you, because this is part of our module in that training, how you can merge these two. But you will not be able to do them together. That is just the end point. It will be challenging if you are doing both of them together, in as much as they are not directional. They're not going towards one direction. They are antagonistic. So you will not do them together. But there is a little way you can maneuver that. That will be stressful. But I will not encourage you to go into that. But you don't do them together. So you just define what you want to do and focus on that aspect. Then the second question, which is on, I think I even answered that question while you were asking. Can you help me, remind me? You were asking about.
Maybe you partially answered it. I mean, those that also have different parameters for scoring.
Now, good, yes. Yeah. Now, I remember now. But those that does not have that 0 to 1 probability score, like maybe TPSA, molar weight, yes, their probability score is not within the range of 0 to 1. So you screen them based on their criteria. Because then we also have criteria. We shall deal on those other ones when we get to pharmacokinetics. Because in pharmacokinetics, we shall also look at the physicochemical properties. So, but okay, we'll still do screening. Yeah, we'll still do screening in the pharmacokinetics. So on that note, because their probability score is not within 0 to 1, you screen them differently according to their own benchmark, because they will have their benchmark. TPSA has this benchmark. Molar weight has its benchmark. But if you're using Excel, you can write a function. See now, you can write a function that can treat them individually. You can write, though the function might be cumbersome, but the function, because what we are doing now, since the ones we are selecting are between 0.1, the function will be very easier. But if the values now, this one, the range is different, the range is from 10, this one range is from 5, another one, the range is from 20, then I will write a function that will treat them individually. I will say, if this is from this to this, I will group it. If this one is from this to this, I will group it. It is possible to develop that function in Excel, since we're not using a programming language to do that. So by assuming we are using programming script, you're using script to do this, it's very easier for you. So I will still encourage us, find a way to learn programming, because of training that will come up later this year ending, or next year, on programming and draw design. So it will be very easier for you. All these things will not be stressful. But we can still use Excel to build that function that can accommodate broad values, irrespective of their differences in probability score. So it is still possible to accommodate them together. You just need to have the idea of the function to write. Once you have the right function, you can do that. Then the first question was, which of these parameters, these toxic parameters, has something relating to reproductive toxicity? Okay, now among these parameters here, none of them has direct impact with the reproductive toxicity. So that is the limitation from this ADMET database. Now, but you can do that, because there is no perfect database. There is no perfect database. So if your interest is on reproductive toxicity, you can do QSAR. You can use that same approach that they used in getting these results. You can do it yourself. That is what we shall do on the 3rd week of February, because the 3rd week of February we have a free training on QSAR and QSPR. So you can now build a model for that reproductive toxicity, that can test your own data. If my data have the tendency for such a reproductive toxicity, so you can build that model. All what you just need is get some compounds and their wet lab results on reproductive toxicity. So the limitation of all this is availability of data. You can go online, you use some Bioinformatics database, get some compounds with known activity on reproductive toxicity. Use it and build a model. At least you can build a simple multilinear regression model, MLR, with your Excel also. Then you can even do optimization with RSM. but MLR is still acceptable. The multilinear regression, you can build it. You just ensure that the model will not be overfitting and will not be underfitting. So once you build it, you can now test your own compound if that compound we have such reproductive toxicity. So you can just keep it aside and wait for us for that QSAR and QSPR, because we are bringing it both for activity and for toxicity. So there is no direct reproductive toxicity from these results. Thank you so much. Okay, do we have another question? I like the question, so it will really help us to be well guided. Okay, so we go ahead, right? Hello?
Yes, sir.
All right. Now, what we'll do now is...
Yes, sir, you can go ahead.
Okay, what we'll do now is we are going to select the data we want. There's something that one of us observed on this data that if you look at this row, the third row has how will I explain? But you can see what is there. Values that we are continuation of the second row. So while you are working with your data, did you encounter this challenge? Did you know if you also encountered this while working with your own data?
Yes,
That's
sir.
for those that didn't. Yeah?
Yes, sir. Yes, sir, I did.
With your own data, not my data.
Yes, with my own.
So it's just a minor challenge. What we just need to do is that if you look at, let me zoom my screen. So if you look at this third row, it's supposed to start with a smile code, but it's not starting with a smile code. That means it could be a continuation from the previous one. So we'll just delete it. We'll just delete, select those ones that repeated. So for me, I'm seeing my row 3, row 11, 17, 21.
13.
So,
13 is there too.
25.
17 is not included, though. You are deleting the smile at 17. No, check it again, please.
Am I having a problem? Okay, yeah, thank you. Oh, thank you for that. Okay, sorry. 3, 11, 13, good, thank you. 16, yes, 16. 18 doesn't have a smile. 21, 25, 27, 29. So I would just, having highlighted them, I will come to delete cells, and it's deleted. So how many left now? How many left? So this is 21, which corresponds to 20, because the data starts from number 2 to 21. So this is what we shall be using now to work with. So in case you encounter such a thing, don't don't be afraid. It's normal sometimes. So let us select our data. Let's select the data we are using to work. Let's select the data we are using to work. So you can still remember those 17. Let me just run it fast with you so that you can still remember, so that it will guide us while we are working. The 17 we want to work with. So we just have it. We are working with genotoxicity. We are working with nephrotoxicity. We are working with hepatotoxicity. We are working with autotoxicity. We are working with neurotoxicity, HHT, respiratory toxicity, eye irritation. We are also working with eye correction. We are working with carcinogenicity. We are working with skin sensitization, FDA, MDD, ROA, then MS, DILI, and the 2H, the head without concentration, the head with concentration. So this will now guide us while selecting. So a quick one. At this point, see what you do. But before now you have merged your other data, like the other ones you work with, you bring them under, you come to this step below and you paste. You keep joining all the data until you have correlated all your ADMET results from your data. So what you just do is come to, let's pick their smiles. So let's take this smiles at column B. So click column B. Click column B. Click column B, or just highlight down to column B. Once you highlight column B, hold your control while we move towards the other parameters we want to pick. While we move toward the ones we want to pick. So you are working with me. Are we together at this point? Hello? Hello, are we together?
Yeah, we are.
Okay. Okay. So what I said is you get your column B, which is showing your smiles, not the raw smiles, the other smiles from column B. Highlight that column B, then hold your control key while you move towards the toxic parameters we want to select for screening. So you look at the toxic parameters you want to select. We are picking H, HDI, MS, IOA, FDA, carcinogenic, ECEI, respiratory disorder, HHT, neurotoxicity, autotoxicity, hepatotoxicity, nephrotoxicity. I'm highlighting them. So let me count. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17. So it's complete 17. I do Control C. So I copy. So you see that the 17 are already grouped. So there's no need for you to stress yourself from HERG down to genotoxicity. So once you copy, you come to down. There is this plus sign which indicates you should add a new sheet. So click on that to add new sheet. Then you put Control V, you paste. So we have selected the data we want to use for screening. So I can just rename my sheet to say screening data. I can say screening data. All right. Are we on the same page?
Yes, we are.
Okay. Kindly do it to ask question if you are not clear. So at this point, we want to screen. You see that it will be stressful if you begin to scroll one by one to see the ones that will pass and the ones that will not pass. It will be stressful. So we are going to screen all at the same time, setting our benchmark. So which benchmark should we use? What should be our benchmark for the screening?
It should be 0.1 to 0.3.
Okay. So let's see if I would prefer using that. That will be very good. So that means we are saying that we want to select data that the maximum should be less than 0.4. Okay, or we say less than 0.31. So since our margin is between 0 to 0.3, so anything above 0.3, maybe 0.31, it should tell us toxic. But anything below 0.3, let it tell us non-toxic. So that is what we're going to use. So that means we will set it maximum value. So we are working on below 0.31, because below 0.31, and that means from 0.3 down. So we shall use a self function. What you just need to do is identify the cells you are screening. We are screening from B2, from cell B2 down to cell R2. So we are screening that row from cell B2 to cell R2. So you come to this space. I will still zoom my screen so that you can be able to see it clear. So you come to this at the next column. We write a function there. We say equal to. We use each function. Okay, no. Let me write screening or validation. Let me call it validation. So I want to see my validation. So that means I'm screening from B2 to R2. I'll say equal to. I'll say if. Once you type if, you open bracket, you say maximum, mass. If the maximum value, you close, you open another bracket. If the maximum value between B2 to R2, you close bracket. So you can follow what I'm doing. If the maximum value between B2 to R2, if the maximum value is less than, if it's less than 0.31, are we together? Hello? All right. So
Hi.
We are together.
all right. If the maximum value is less than 0.31, because we are working with to be less than 0.31 is from 0.3. So if the maximum value is less than 0.31, comma, what do we say? Non-toxic. It should tell us non-toxic. It should tell us non-toxic. But if it is, if the maximum value is, if any of them, because we are combining these toxic parameters at once. So if any of them, even if it's one out of these 17 parameters that has a maximum value greater than 0.3, let it read for us toxic. Let it read for us toxic. You put your question mark and you close brackets. So did we got the function correctly?
Yes, I think so.
Then enter. So mine is saying for the first one, toxic. Then you can drag down. You can drag down. So glory be to God, all my compounds are toxic. I don't know about yours. So check yours. So I want to get your feedback.
Hello,
Check
sir.
yours.
Hello, sir.
Yes.
Okay, if all of them are toxic, it means they are not good now.
Yes.
Okay, so why the glory be to God? Okay.
Of course, now I'm grateful. That means I should be careful working with those compounds.
But sir, I didn't quite get the validation formula clearly. You said less than 0.31 non-toxic.
Yes.
And then you now wrote toxic. You didn't say more than 0.31.
Okay. Yes. What the equation does is that is like otherwise. So
Okay,
the question, the equation understands otherwise. So anything not below 0.31, it will just read
okay,
it toxic. Yeah.
okay, okay. Anything more than 0.31 will now be toxic. Okay.
That be toxic.
Thank
Yes, because from the class from the training this evening, we able to see that from all these parameters, their benchmark, good appropriate benchmark are 0.0 to 0.3. So except you want to extend a little bit, you want to extend to 0.5.
No, sir, it's okay. I just wanted to understand why you did not imply more than 0.31 toxic.
Okay.
But you said that the command understands what you're saying, so it's okay.
Yes.
Thank you, sir.
All right. Okay.
Well, sir.
Yes.
Thank you for this presentation. I want to inquire or maybe submit that some of these drugs, those drugs that particularly chemotherapeutic drugs, they do have some side effects.
Yes.
Now, so despite that those side effects, there is no alarm for the withdrawal of those drugs from market. Then all these our natural product too, and then also not scaling through. Probably since this one is the computer aided approach. So by the time we now do the in vivo, possibly they can still navigate their way and become a reasonable drug. Are we not seeing from that angle too?
Okay.
Or we just assume that all the compound that is studied, we just assume that they cannot be good. We just leave the compound and doing further things. That is one.
Okay.
Number two, there is still this. I don't know. Is it part of this all these data? I mean, how do I put it now? All these nephrotoxicity that we are looking at. There is one that they call pains alert, pan-assay interference of a thing. Is it not possible to consider them at this level, at this screening level, to see whether they equally have some structure that can give a false positive results? Moving ahead.
Okay. Yes. You can even, in fact, based on what you just said now, you can combine results from different databases. You can combine results from different databases. So if you want to do a comprehensive work now, like pains alert comes suicide mail. Suicide mail gives that pains alert. So, but Admet Lab does not. So you can now combine the toxic red flags from Swiss Admet, the ones from Admet Lab, the ones from Potos too. We have Potos. So if you, and there are more. So if you can have access to these databases that are specifically for toxicity, then you now check the one, maybe the one that database A has, but database B does not have it. You pick that one and join. The one that database B that C has, maybe the one that is reported in that database C, but is not reported in database A and B, you join them too. You join that one, so you end up having a large toxic scheme for you to work with. So that is what I will encourage you to do. You can exploit other database. The fact remains that in a training, even if it's a 1 year training, we still can't give you everything. We still can't give you everything. We expose you. The exposure can now take you far. Can now take you far to like I told you people, I have a repository that has so many databases. So you just choose the one you want. It can just search online even and set database for those cities, and you see a lot of them. You just need to learn how to work with them. Then coming to the second question, or the first question, which is, if in silico screening confirms that all our compounds are toxic, and we know that in the market there is no drug that is not toxic, there is no drug without side effects, it is because there are drugs with side effects and yet approved. That is why we are fighting, raising alarm for drugs with minimal side effects. And if you watch some of these drugs, Some of these drugs that were approved years back, some of them now are facing withdrawal. You begin to ask yourself, but they approved this drug long time ago, but now they're now withdrawing the drug. Maybe they did not really do surveillance very well on a large population, and now there is reports, and the report is getting too much, so they have to withdraw those drugs. So I believe that very soon too, all those ones that we have in mind that are toxic, that have severe side effects, but yeah, they are still selling, soon they will withdraw them. But at the same time, there is no drug without side effects. We are just preaching for minimal side effects. So what do we do on that note? Should we abandon our plants? No. No. You can go ahead, but you must validate the results. So that is why, if you check among these toxicities, you can now check which of them is really more toxic, like which of them has biology, they will say, has more reoccurrence, has a high prevalent rate. Among the 17, which one really gave high values? You can now go back to the lab and validate the results. So maybe from the results, you're able to see that nephrotoxicity was highly toxic across all the compounds. And this is very important. You can now go to the lab with your formulation, with the natural product powder, or whichever way you want to work with it. You now go back to the lab and validate this toxicity. So you don't rely on in silico results. We always say there is a need to validate your in silico results. Just as we say that it's not possible that they might maneuver it. So the possibility is slim, but validate your result to be sure if what you have in silico is what is reported. Because most times those people that have built this model, that this is a model that we are using, so we are not even sure of how accurate this model they built is. That is why we'll be teaching you how to build your own model, create your own model, so you know if you did the perfect work for yourself or not. You know if the model you built for yourself was right. We are relying on what has been built by people. So, but we are now teaching you can build your model. You can with the help of QSAR and QSPR, machine learning and the AI-driven tools, you can now build your own model for yourself. So you'll be sure if your result is okay. So don't throw away your plants. Continue. Because even at this point, let me just tell you now, at this point, what we did was we mined bioactive compounds. This may not be the bioactive compounds in your own formulation. This is just the bioactive compounds from database, giving us a headline. So where you conclude properly is when you have your formulation, then performed characterization without your formulation. That means whatever result you get from your formulation is what is in your formulation. So if you have 1,000 compounds now in this plant, it may be end up that in the formulation they may not even, most of them may not play a role, may not be feasible in that in my formulation due to a synergistic effect. So, but what we are doing is that while we say screen as much as possible, is giving you headline that this plant has good potential and is good to go ahead with. So it will make you to know if this plant will need my attention more based on the high toxic profiles. But now, even as we have developed this validation, You know, like I explained, out of 17 parameters, only one thing might lead to this toxic report. Hope you know that. Because from the model from the function we created, we are saying if anyone out of these 17 has a value higher than 0.31, say toxic. So let's do another work to count. Let's do counting. Let's count. Let's count among these 17, which of them actually gave, which of them we are more than 0.31. I don't know if you're getting my point now. Are we together?
Yes, sir.
So we can count. So the counting might even help us in making proper decision. Because you won't tell me that in a class, we have only one bad student out of 30. Okay, out of 17, there's one bad student. We'll not conclude that all the students are bad. So let's count how many bad students are in that class. The function just summarize everything that once there is a bad student among the 17 students, then all are bad.
Daniel.
So, but the counting now
Network.
the counting can now count how many of the parameters are toxic. So we can now decide, let's work with the ones that does not have much toxicity based on the number of counting. So I think that will be okay to still help you in making decisions. So how do we do that? How do we do the counting? It's still Excel. It's still an Excel function. You say equal to, you write count if. Count if. That is the function, count if. You open brackets. What are you counting? I'm counting from B2 to R2, comma. Count those ones that count anyone. So let's say count anyone that is higher than 0. Okay, count how many less than 0.31. Yes, count how many less than 0.31. Okay, are we together?
Yes, sir.
So we want to count. This counting say from my B2 to R2. Tell me out of this 17, how many is really less than 0.31? So how many that pass? That is by saying less than 0.31. How many that passed? Why, if I turn it greater than 0.30, it will tell me how many that gave birth to that toxic report. So let's concentrate on how many that are good out of this 17. So I'm putting less than 0.31. So once I hit enter, something is something is fishing here. Do I need to put something?
Close the R2 bracket.
The R2
bracket, yes.
Okay. I don't think so. Well, let me see. No.
And that comma after the bracket.
It should not be there.
It should be there.
I have to look for. Okay. Okay. This should be, sorry. This should be this. Let me see. This should be. I just want. All right. Okay. So. Okay. So this is the right function. Sorry for. Just have to rearrange myself to make sure we get it. So this is the right function. This is the right function. Count if my B2 to R2 on this my criteria of 0.31, how many will be less than 0.31? So if you do that, here it's telling me 10 out of 17. So we can now judge. I believe most of us are already in this, we're in the academic field. So 10 out of 17, should it go? So what will be our criteria now for this one, for the counting? What should be our pass mark for the counting? Over what? Yes, let's decide what should be our pass mark for the counting.
Let's go conventional, 50%.
Okay. So, 50% as a pass mark. That means one people to pass your course. Everybody will pass your course.
Is that not generous?
That is too generous.
Let's make an A then, 75%.
75%. So
if any of them will pass.
Okay, 75% of 17 will be how many? 0.7 times 17.
That's about 12 point something.
So about 11.9, so 12.
Yeah.
So those that stop to 12 can go. Those, I know, this is what some of us do. When we have when we have marked our script and we see that this people did not do well, you will now try to be a bit flexible. But most times it doesn't help matter. It's just for the sake of our HOD not to say, please help these students. The failure is too much. So let's sort it. Let's sort it so it will be easier for us. Let's go to data, go to sort. So let's sort that last column so it will be easier for us to really, because assuming they are just 20, so we can easily count. But if they are too much, so let's say from smallest to largest or from largest to smallest. Okay, so if you are keeping a benchmark at 12, what can we see now? So what is the implication now if we keep our benchmark at 12?
One say true.
One say true. So you see that the counting function, you can rely on it. You can rely on it instead of you just concluding all toxic. You can look at, okay, that this simply means that out of this my 17 parameters, 12 stay true. And if you carefully count it, you can identify those 12. You can identify those 12. This is 1, this is 2, this is 3, this is 4, this is 5, this is 6, this is 7, this is 8, this is 9, 10, 11, and the 11 and 12. So this is the 12. So you can look. In fact, there's another function that will even help you to highlight those ones that are toxic or non-toxic, like those parameters that we are more than 0.31. There are so many functions that you can manipulate in doing your screening. There are too many I use them too many. There are many. You just have to go online, search, and some of the things I use, I didn't learn any of them when I was learning. When we had trainings like this from years ago, most times they are just from your exposure and your experience in data analysis. Then you apply them. I will even encourage some of us to learn data analysis, Tableau, SQL. They will help you to do all this analysis for you. Python, R programming, you can learn them. Yes, once you are out for research, there are a lot of things that you learn. There's a lot of things, like some persons has already signed up for a 12-month training, both paid and free. So I was like, do you have purpose? Or well, to me, I would say that the person has enough money, but that is maybe the person has a purpose. Just sign up. So it's automatic. Add me to all training, both the paid and the free webinar. Just add me. So that stress of me filling form, just add me to the group chat once you trade. It's okay. It's even what is good. So such a person now wants to just use this year and gather all the training that are possible. But please, while you are training, navigate. As you are learning, begin to look at the possibility of what you are learning. It's not just to acquire, and you just acquired and no application. You have not utilized it anywhere. So that is not what we preach. So I can say that at this point we are through with our toxicity screening. So save this data, save this result. You can save it as, it was as a CSV. You can save it as an Excel file. You can save it as a workbook so that it will be very easier for you to work with. So you can save it as an Excel workbook. Then I'll call it natural product screening, or I call it toxicity screening. So whichever one you want to call it, you can call it toxicity screening. So if you want to now continue, you continue with this. For me, I can say 10. 10 out of 17 is what percentage? 10 divided by 17 times 100. That is 58. And that's only 60%. So I can equally work with 10. I equally work with 10 because it's still good. But then, please, there are some toxic parameters that we must be careful with. Mostly all these ones that affect organs, like the respiratory, the liver, the kidney, the eye, the blood, the genome and cancer. All those ones, you should be very careful with them because FDA usually focus on those ones. Although since we are developing herbal products, we may not need FDA approval in Nigeria because NAFDAC can take off that. But when you are now exporting to other countries, then you need that proper FDA approval. But with NAFDAC approval, you can start selling. And we know how it is in NAFDAC approval. But we will not take because of that, and we will not do a good work. There if you screen some of these products in the market, these herbal products in the market are, if you screen them, you actually marvel what people are doing just because they want to sell. But even if you want to sell or want to treat, we'll just follow the right procedure. So at this point, let me take questions as we summarize for today's training. Let me take questions. Okay. Sorry, Prof. I this is your area, Professor Justin. On board. So this is your area, and I would have even left you to handle this session.
I'm around, sir.
I would have allowed you to anchor this session because this is your area. I can remember.
Yes. I'm learning. I'm still learning, sir.
No. Yeah. You're not learning again. You are both learning.
No.
Once again, I want to appreciate you. So now we are getting closer. So what is remaining for us now is about how many modules? 4 modules. The lead mining, which is the natural product mining, the lead mining, the bioinformatics, the pharmacodynamics, and then pharmacokinetics. So, but we'll see how we can enshrine this to cover up between now and next week. Between now and next week, let's see what we can do. We have about 3 more or 4 classes. So let's see what we can cover up within that period. I really want to say thank you for being around. So we have questions. You have questions. Questions, suggestions, and your opinion. So while we wait for questions, then please, I will always encourage you to be doing your work. Do your work. So at each stage now, I don't think if there is assignment again. The assignment is now your work. So do your work. At each stage you do. Now that we are done with natural product mining, with toxicity screening, that implies that by tomorrow when you settle down, you can try to do something with your data. Then when we resume on Friday, we shall now look at identifying leads from those compounds that passed, or maybe from still all the data, identifying lead compounds from there. Then once we have identified the lead compounds, still using screening, then we'll now go to our bioinformatics. Yeah, we'll now look at the bioinformatics. Of course, once we entered bioinformatics, we are done with the work because we are now entering our normal doctrine. We entered our normal doctrine, so we expose it to some bioinformatics database, which we may not go deep because bioinformatics is a course on its own. Is something else on its own. We shall just look at genomics and proteomics, and then some of their databases. So keep working with your work. Keep working with your data. Keep working with your data. Very, very important. Keep working with your data. So once again, I'll say thank you for being around. Thank you so much.
Sorry, before you go, to follow up to my question for the reprotoxicity, can you suggest any database I can use for that?
Okay, I will drop some of them in our WhatsApp platform. I will recommend some so you can exploit them.
All right, thank you.
Yeah, you can exploit them. So it will help in making your work robust.
Hello, sir.
Yes,
Okay,
Prof.
sir, please, when you were talking about the count, I just want to know the criteria for the count and why. What are we actually counting? What are we looking out for? After the toxicity screening validation, the count,
Yeah.
what exactly is it for? Please, can you clarify me?
Okay. Yes. We have a class of 17 students. And in that class of 17 students, we wanted to see the students that are good. That's our idea. We had 17 toxicities, and we're looking at the probability of a compound to pass these 17 toxic profiles. So let me to have a class of 17 students, and we are not looking at the probability of the number of students who are good. So I'm using good for the ones that will pass, which is below 0.3. Then I'm using bad students for the ones above 0.3. So in the screening, we screen the 17 students. And in the screening was saying that once there is a bad student in that class, then we'll term all the class, we'll term that class to be bad. That was what the screening does. So once there is a student that the value is above 0.31, the screening will just give us toxic, bad. But we are doing that without actually looking at why not we check how many of these students are actually bad in this class. Because it's unfair that you just come to maybe because one lecturer is bad in the department, you will term others bad. And you need to count which ones are bad. At least let's know to what extent, to what percentage we are there actually bad. So let's know. So that was why we wait for counting. So we are counting how many bad students are in that class or how many good students are in that class. So we focus on the good students. Since the presence of a bad student will term the whole class bad, let's actually count how many good students are there. And maybe we can now set a criteria for percentage of good that we can say, if you have so many of good students, it's just like what happened in the Bible, in the Holy Bible, between that is in Sodom and Gomorrah. Yeah, so Abraham has to bargain. What if you see so number of person, will you still destroy? And that is what we want to do. We want to bargain. So in the count, we are counting how many good students are in that class. So you can see that the result gave 12, 10, which is those values are how many of these parameters we are below 0.31. So those ones, how many of them from out of 17? For each compound, how many we are below 0.31? He gave the function have to count it by itself and say 12 are good. That means 12 are below 0.31. 10 are good. Then when we're able to get that, you now being a researcher, you are a researcher. You cannot develop your criteria. Like one of us was saying earlier, 50%. I disagree that 50% is too poor. 50%, 50 that is half of 17. So it was like, let those that have up to 9, let's call them pass mark, from 9 pass mark, which to me is generosity. we said 75%, which I agree to. I agree to that, because in your studies, in your school, 70 is A. Depending on your school, 75 is A. So let's work with that criteria for A, that criteria for excellence. So you can now decide to work with 70%. So out of 17, what is 70%? You say 0.7 times 17, we got 11.9, which is 12. So you can decide to work from 11 up, 11, 12, which now depends on the number of your toxic parameters that you're considering. So 70% is okay for your count. 70% is okay for your count.
Okay, sir. Thank you, sir.
Yes. Thank you very much.
So, sir, we're using all of these 17 parameters for our own work, right?
Yes, but if I provide other database, you can exploit and expand your own. Is that possible?
I don't know for now. But give us the database special.
If you can, it's just if you can. But if you see that it's not possible, then just limit first for this 17. This is a learning process. This is just a learning process. If you are doing your work like a work, what today you can now take time, absorb everything we have done and including it. So this is just a learning process. You don't kill yourself in learning process. You just try your ability and see that it works.
Okay, sir.
Okay, thank you so much. Okay, once again, I want to say thank you, and it's 3 minutes to 10:30, so I can make up for my time. I appreciate your presence. I really appreciate your presence, and the training has been, to me, I'm learning a lot from the training, because your questions, your response, your opinions, and your suggestion is really providing loopholes for us. Some of the challenge, and is making us to see the future. See the future. So once again, tomorrow, I will be meeting with some persons here tomorrow on the beginner's course on computational chemistry. That training will be wonderful. It's starting tomorrow, and then February on green chemistry is already at the corner. We have already released the advert for chemoinformatics. I'm trying to see how we can exploit 10 database in 2 days. Just expose you to 10 database in 2 days, show you what they do, and that is actually my target in that chemoinformatics. So help us spread it. The advert actually came late because the training is starting next week. So just try, let's see how we can spread it around our friends and police and students. Chemoinformatics is another aspect that is not neglected. Once again, thank you and God bless you all. See you on Friday.
Thank you, sir.
Thank you. Thank you.